Okay, sexual reproduction in flowering plants. First of all, let's understand why sexual reproduction is a requirement for an organization or for an organisms. What do you think about it? To produce offspring first to the next level, and also to develop the DNA, okay? Developing the DNA and producing a better offspring from what it was being produced before. Okay, and there is an evolution also happening, isn't it? So each and every organisms which exists in the world has a specific purpose for it. So in that reproduction, producing new offsprings, it's really fascinating how it happens and why it happens. So what's the rule, the God's rule for this reproduction? What kind of reproduction happens? What is a bisexual flower and how it will be useful? Why there are flowers? Why there are flowers with scents? Why they are producing honey? And what is the purpose of stamens? What is the purpose of thalamus? What is the purpose of leaves? And how we are connected to it? All other living organisms, animal kingdoms, or anything. How each of us are connected to this. All these are a fascinating study. When you understand it, it will be a miracle. We ourselves are a miracle. All right. An egg has been produced once in every 28 days, and almost crores and crores of sperms fight with each other to produce it. So we ourselves are a miracle. We fought with crores and crores of sperms to get to this position. So we are miracle ourselves. A lot of people think that some people are celebrities, they are this, they are that, but each and every person is a celebrity. They can arise to that position because they are a fighter from the start. Okay? So that's why we are going to work on this. What and all we are going to work? Flower, a fascinating organ of angiosperms, pre-fertilization, structure and events, double fertilization, post-fertilization, structures and events, apomixis, polyembryony. Okay. Actually, we went through the notes, but we have to study the whole book first. After that, when you go through the notes, you will understand before going to exams or everything, it will be easy for you.
Actually, most of the questions are going from the textbook only.
All right.
Each and every line.
What we would be doing is, we would be having a specific time limit. After completing each and every part, if you are completing this part, we'll be discussing about that, but what you feel about that, and if you want to add some more to it, we'll be adding. Then the second part, the flower, a fascinating organ of angiosperms. Okay. Then pre-fertilization structures and events. Thus, after completing this, we will be having a discussion. What kind of questions can be asked from these first part, 2.1? Then pre-fertilization structures and events. What is a pre-fertilization structure? How we can relate to it? Stamens, microsporangium, and pollen grains. What are these? Actually, we went through all these beforehand, but to ensure and emphasize the importance of this, we will go through it again and again. It's not a problem. Okay? Each and every part. Before going into that part, we'll be seeing what and all. What and all. You should remember, we explain all these, and also we went through these things. Do you remember this? One by one we'll be proceeding. Don't worry about it. One by one we'll be proceeding. There will be a lot of questions from this part. The pollination, third part. So it's a big lesson. When we consider other lessons, the sexual reproduction in flowering plants is a bigger lesson. But when you understand it, it will be very simple. Let's start. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and perfumes that we swoon over, the rich colors that attracts us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of the inflorescence, flowers and floral parts shows an amazing range of adaptations to ensure formation of an end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants, angiosperms. What is this? Myris. Inflorescence. What is myris? What is myrids? Different groups of flowers which we enjoy. Different set. Suppose take a bouquet. There will be arrangement of different flowers in an order. That is known as myrids. Okay. What is an inflorescence? Inflorescence. What is an inflorescence? Mo, what do you think about inflorescence? There are simple single flowers. There will be a bunch of flowers in a single stalk. For example, when you think about party, a lot of flowers in a single stalk itself. There is two types of, yeah, you have lots and lots of inflorescence in your gardens too. Hmm. No, inflorescence are of different types, racemose, zymose. Even a sunflower is an inflorescence. The outer petal is a male flower, and the inner one, the black one, which we see, it is the female bunch of female flowers. It's not a single flower. A single flower is hibiscus.
Can be both also, right?
Hmm?
Can be both also, right?
Which one?
Hibiscus.
Hmm.
Can be both, right?
Both. I couldn't get it. What you're saying?
Hibiscus can be both, as male and female reproductive.
No, no. We are not speaking about male bisexual flowers. Inflorescence, a bunch. See here, like this. In a single stalk, there will be lots and lots of flowers. A bunch of flowers in a single stalk. This is known as inflorescence. There are different types of inflorescence. Even even a sunflower or a, see, this is also an inflorescence. You have a mango tree in your garden?
Yes.
Have you seen the flowers of mango tree?
No.
Small, it will be very, very small. It will be in a bunch. It will be in a bunch. Why we are getting lot of mangoes in a single stalk, a bunch of mangoes? Because the flowers which are getting pollinated alone would become, there'll be numerous flowers. And that some only would get pollinated. Those would become mango fruit. That's why we have in a single stalk itself, we'll be having a bunch of mangoes hanging there. Okay. So this is known as inflorescence, a group of flowers. All right. This is known as inflorescence. Okay.
Just take a note.
Okay. Okay.
I hope nobody snatches this also. They won't hesitate actually if they see this.
Flower, a fascinating organ of angiosperms. Human beings have an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated in our homes, and in gardens. Find out the names of 5 more flowers that are used in social and cultural celebration in your family. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. In class 11, you have read the various parts of flowers. Fig 2.1 will help you recall the parts of the typical flowers. Can you name the 2 parts in the flower in which the 2 most important units of sexual reproduction develop? What is floriculture? Floriculture. What is agriculture?
Harvesting of flowers.
Okay, then what can
Harvesting
be
of
floriculture?
flowers.
Yeah, they will be having a separate farm, not a small farm, a big farm. Floriculture. Good. Okay. See here. This is the simple flower, isn't it? Simple flower. All right. And the simple flower, this is known as thalamus. This is known as thalamus. In thalamus only, the innermost, this part is known as sepals, calyx. 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 This is petals, also known as corolla. Corolla. And male reproductive organ, stamens. Then female reproductive organ, gynoecium. This totally. All right, gynoecium. This would be having the pollen grains. Here is the ovary. The pollen grains would get deposited in stigma. It is sticky for pollen grains to get settled. known as stigma, and it produces pollen tube. The pollen grains will be producing pollen tube, and this will come and meet the ovary here, and from the lower part it will fuse with the egg. Okay. This is what happens. We are going to look into it in detail, what would be happening and why it's happening. Pre-fertilization structures and events. Much before the actual flower is seen on the plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated, which led to the differentiation and further development of floral primordium. Inflorescences are formed, which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and gynoecium, differentiate and develop. You could recollect that the androecium consists of whorl of stamens, representing the male reproductive organ, and gynoecium represent female reproductive organ. Stamen. There will be lots and lots of questions from this, all right? Before looking into this, we have to look at the pictures and compare it with this, all right? This is the stamen. This is on a stalk, anthers, and when we cut the anthers, we'd be having two lobes. Each lobe would be containing two locuses, all right? Dehiscence. Dehiscence means where it breaks, all right? You'll be having this dairy milk chocolate, isn't it? No chocolates. Any chips or anything when we are opening.
Chocolate.
Hmm?
Dark chocolate.
Whatever chocolate, when we are opening that cover, there will be a specific place where we can easily open. Correct? That is known as place of dehiscence, where it breaks and opens up. This is where it would break and open up and exposes the pollen grains, disperses the pollen grains. Then we will be looking deep into the structure. We'll be looking deep into that structure. If we cut the stamen, it would be looking like this. And when we expand, there will be 4 layers. We'll be looking into this. Then how the pollen grains have been produced inside, how they will mature. That's where the pollen grain ends. All right, production. Then after that, we'll be explaining what is the structure of pollen grain, how it would be useful. Okay. Stamens, microsporangium, and pollen grains. Fig 2.2a shows the two parts of typical stamens, the long and slender stalk called the filament, and the terminal, generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen, each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobe, with each lobe having two theca. They are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobe nature of an anther is very distinct in transverse section of the anther. The anther's 4-sided tetragonal structure consists of 4 microsporangia. located at the corners of two in each lobe. The microsporangia develop further and becomes pollen sacs. They extend longitudinally all through the length of the anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers: epidermis, endothelium, middle layer, tapetum. The outer 3 walls perform the function of production and help in dehiscence of anther to release the pollen grain. The innermost layer is called the tapetum. It nourishes the developing pollen grain, Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? How it can become binucleate? Is there any option which you can see? Binucleate. There should be 2 types of division: mitosis or meiosis. Which one it would be?
No.
Meiosis is for sexual after the formation of embryo.
Formation.
Generally, for the growth and everything, mitosis would be the fundamental. Okay. Okay. When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each sporangium. Microsporogenesis. Microsporogenesis. What is microsporogenesis for you?
Small.
Genesis itself is the development.
Developments.
Okay? That is how the pollen grains are getting developed. The process of producing pollen grains can be known as microsporogenesis. If it's megasporogenesis, the process of forming ovary from the mother cell, how it forms pollen grains. All right. So as the anther develops, the cells of the sporogenous tissue undergo mitotic division to form microspore tetrad. What would be polydy of the cells of the tetrad? See here, this is sporogenous tissue. There will be 4 layers. First 3 layers, epidermis, and the endothelium, and middle layer. These are for protection. Okay, middle layer. These are for protection. And the tapetum alone, which is the almost the inner layer which surrounding the sporogenous tissue. All right, that alone will nourish the pollen grains. It will help in development. All other 3, the first 3 would be for protection and dehiscence. All right, protection and dehiscence. Okay, check here. So epidermis, endothelium, middle layer. After that, tapetum. Tapetum will be helping. This tapetum would be helping the. This tapetum would be helping the formation of new. It will be nourishing the pollen mother cells. What happened? What happened? Huh?
1 minute, I washed my face.
Okay. Okay.
Suddenly, I don't know why.
Okay. Okay. So this tapetum will be nourishing the pollen mother cells. All right. We'll be looking into that. Don't worry about it. So as it gets nourishes, This will dehisce and produce the pollen grains outside. So, as each cell of sporogenous tissue is capable of giving rise to microspore tetrad, each one is the potential pollen grain or microspore mother cell. So, sporogenous tissue have microspore. This microspore, each and every microspore can be converted into pollen grains. Microscope, potential pollen grains. That is also known as microspore mother cell. All right. The process of formation of microspore from pollen mother cell, PMC, pollen mother cell, through meiosis is called microsporogenesis. Right? That is one important question which you may get.
I just saw a question about it.
We'll be getting a lot of questions, don't worry, regarding this. This chapter is one of the most important chapters. Lots and lots of questions can be asked from this.
I don't think it's like that. Most of the questions are coming from this also it's coming, but most of the questions are coming from the next chapter.
They are also very important. We'll be getting a lot of questions, don't worry. We will proceed.
Because I don't want to do that same mistake again.
Okay. The process of formation of pollen grains from pollen mother cell is known as microsporogenesis. All right. The microspores, as they are formed, are arranged in a cluster of 4 cells. The microspore tetrad. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released with dehiscence of anther. Okay. So, since there are 4 lobes, it is known as microspore tetrad. And as it gets dehydrated, whatever.
I'm taking each and every detail.
Here there are 2 pencils.
That one is almost dead. Oh no, I thought I lost this. Okay.
Okay. Now we are going to go into detail regarding pollen grains. Like we already discussed, we have to look into the pictures before proceeding into it. See here, different kinds of pollen grains, and in each and every, yeah,
this is COVID.
no, we discussed and told that it looks like COVID virus. At the time we discussed,
Yeah.
And we thought, oh, this looks like a COVID virus.
Actually, it's not. It's not.
At the time, COVID was in a very strong situation, isn't it? So that we discussed.
Supposed to come like this, somewhat like a stem and then like this.
There are different variations. See, look into the walls. Look into the walls of each of these diagrams. When we focus, there is a small opening like this, and this one pollen grain can be converted into how we are getting into this. When it matures, what happens? Okay. And slowly it will become dinucleate. After the synthesis, this one would be the generative cell and this one would be the vegetative cell. This nourishes, which helps in formation of pollen tube and how it's going to find the embryo. All right. So vegetative cell and generative cell, it will be dinucleated. We are going to read about all these now regarding pollen. It's very, very important regarding this. This
But they
whole,
won't ask questions about that.
what? They asked already. I saw in those questions.
I mean, they've asked, but not much.
It won't be taking more time. It's better to move in clearly. Okay. The pollen grains represent the male gametophytes. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under the microscope. You will be amazed by the variety of architecture, size, shapes, colors, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometer. Micrometers 10 to the power minus 6. Right, 10 to the power minus 6. It has a prominent two-layered wall. The hard outer layer called exine is made up of sporopollen, which is one of the most resistant organic material known. It can withstand high temperatures, strong acids, and alkali. No enzyme that degrades sporopollen is so far known. Pollen grain exine has prominent aperture called germ pores, where sporopollen is absent. So from the germ pore only, pollen tube is formed. There will be a small opening in exine, outer shell. From this only, pollen tube would be formed. Pollen grains are well preserved as fossils because of the presence of sporopollen. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of germ spore? The inner wall of the pollen grain is called intine. It is a thin and continuous layer made up of cellulose and pectin. Numerous times they have this question. What is exine made up of? It's made up of sporopollen, all right, which is one of the most resistive enzyme layer. It will not be affected by high temperature or or acids or alkali. Nothing will affect this sporopollen. We have a germ pore. From that only we can get the pollen tube, or else it will not be working at all. Then regarding the inner layer, the inner wall is known as intine, which is made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. The vegetative cell is bigger, as abundant food reserves and a large, irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and nucleus. In over 60% of angiosperms, pollen grains are shed at this two-cell stage. In the remaining species, the generative cell divides mitotically to give rise to two male gametes before pollen grains are shed, three-cell stage. Almost for 60%, it will be in two-cell stage only. The vegetative cell will be large and irregular in shape. The nucleus is also will be irregular. The generative cell, which contains the DNA, which will be small and it will be moving inside the cytoplasm, small and surrounded in the vegetative cell, which will be inside the vegetative cell. Pollen grains of many species cause several allergies and bronchial afflictions in some people, often leading to chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a containment with imported wheat has become ubiquitous in the occurrence and cause pollen allergy. Have you seen this Parthenium? Parthenium.
Oh, carrot grass I've seen.
Parthenium, carrot grass name is Parthenium. All right.
It's a scientific name.
Yeah.
Okay.
Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets, syrups are available in market. Pollen consumption has been claimed to increase the performance of athletes and horse race. Rich in nutrients.
Only for the horses.
Yeah, for running faster and faster, it needs lots of energy, isn't it? If you run faster than what you do,
I thought only for us. I didn't expect horses.
It has been given for horses too.
Okay.
Pollen bee pollen from England. See, there are creams, energy shakes, syrups from Poland.
I'm actually planning to buy one in the future.
Get it, so you become really active, energized.
Definitely, because I'm already very weak. I lost my running speed.
When once they are shed, pollen grains have to land on the stigma before they lose viability. What is viability?
Their quality.
It's not like quality.
Like
their the reproductive
ability,
strength.
I meant to say.
Ability, yeah. All right, ability, reproductive ability. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depend on prevailing temperature and humidity. In some cereals such as rice, wheat, pollen grains viability within 30 minutes. Rice and wheat within 30 minutes. And in some members of Rosaceae, Leguminaceae, and Solanaceae, they maintain viability for months. You may have heard of storing semen or sperm of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen at minus 196 degrees Celsius. Such stored pollen can be used as a pollen bank, similar to the seed bank in crop breeding programs. Okay. Now, we went through a lot of things in pollen grains. All right. Can you give a briefing about what and all you went through in pollen grains? From here, it would be good. Or, yeah. What and all you remember?
Inflorescence means a group of flowers.
It can be like, for the right word would be bunch of flowers.
Bunch of flowers.
All right.
bunch of flowers, and flowers can be bisexual. They have both male and female reproductive part.
Okay.
Flowers can also be unisexual.
Yeah, can be unisexual. What do you remember about microsporangium? Stamen. Look at
That
the
they
picture and
contain
explain.
they contain the wall. They contain many layers
Okay.
which protects the like for they have four wall layers.
Hmm.
Epidermis,
Hmm.
endo
Endothelium.
endothelium, middle layer, and
Tapetum.
tatum. The innermost layer is actually tatum, and the outermost layer is epidermis.
Epidermis.
Mhm.
Okay. Then I will be asking questions regarding it then. What are the main parts of stamen? There are two parts. This one and this one.
Anther and filament.
Anther and filament. Long stalk, which is attached to the thalamus, is the filament. Anther, which produces the
Anther, which produces the
pollen
pollen
grains.
grains.
Okay, pollen grains. It is two-lobe dithecous. So there will be four
structures. Four
which can be
pollen sacs.
pollen sacs, which can be dehisced in between. Dehiscence can be, there will be a groove for dehiscence for it. Okay. So when we cut it transversely, we can see that the anthers are four layered. All right, four layered. Epidermis, endothelium, middle layer, tapetum.
Tapetum.
Okay, tapetum. Out of this four, first three is for protection and dehiscence, dispersal of pollen grains. The inner one, tapetum, which nourishes the microspore, and it helps in development of pollen grains. Tapetum. All right. Okay. Then come here. So this is how it looks. As the pollen mother cell matures using the nourishment of tapetum cells. Tapetum have a dense cytoplasm. All right, dense cytoplasm. With that, it will get nourished. Each and every microspore can become a pollen grain. All right, can become a pollen grain. When it dehydrates and dehisces, it disperses or liberates the pollen grains. All right. Now we are coming into pollen grains. Pollen grain. The kind of example which they gave us hibiscus. If we rub the hibiscus flower, that stamen, it will be
yellowish powder.
Yeah, pollen grains will
be
be formed. If you look into it, look that
There'll
in a
be
slide.
different shapes,
Yeah, different
color,
size
and design.
Generally, pollen grains is of spherical shape and very minute. Its diameter is around 25 to 50 micrometers. Okay. It is a two-layer. It is two-layer. The outer tough layer is known as exine.
Which is made out of
Sporopollen. Sporopollen, which is the most resistive organic material available. It doesn't get affected by acids, temperature, or alkali. There is no chemical which we have found out such a way to disintegrate the sporopollen. It has a, it has a small hole, a pore in it, known as germ pore, which doesn't have sporopollen. The purpose of germ pore is, from that only we'll be getting the pollen tube to move the male reproductive nucleus to the embryo. All right. Okay. The inner wall is a thin layered wall known as intine, which is made up of cellulose and pectin.
It is a thin and confused layer made up of cellulose and pectin.
As the pollen matures and disperses, it would divide mitotically to form two cells. One is vegetative cell, which is large and the nucleus is irregular, and the other is generative cells, which is small and it is inside the cytoplasm of vegetative cell. Okay. Generally, 60% of flowering plants have two cell stage like this, and remaining may have 3-celled. Okay, this is what we found out in this. This sporopollen is rich in proteins. So in developed countries, they are using this pollen grains to form pollen tablets instead of next page. It will be in next page. Pollen tablets and syrups and milkshakes, shakes and everything, so that it is good for it. It is highly nutritious and were taken by athletes and given to race horses too. All right. The viability of pollen grains differ with species. The viability of pollen grains, the strength of the male gamete inside that pollen grain would be having a certain time limit after the dispersal. All right. That will be varying. For grains like wheat, cereals, it is 30 minutes. Within 30 minutes, it has to go and find the stigma, or else there will not be any pollination. It will not be of any use. The pollen grain will be dormant. It will not be any use. Like the sperms are getting stored for artificial insemination, pollen grains also can be stored. All right. So that we can preserve some species, isn't it? There are a lot of species which are getting extinct. So if we could preserve the pollen grains, it can be used to produce seeds and regenerate the extinct species. All right. See, after completing this, I went through the whole one by one. Okay, what's this? What's this? What's this? What's this? We don't need a book. It is a continuous process. It is a continuous process. The way to read biology is not like these are the questions. It's a continuous process. Whatever question is being asked, you can easily answer it. Okay. The thing is, you're getting pressurized because of this exams, schools, and everything together. It is just like a story. What happens, and what happens.
Especially how my ma'am always gets irritated and sleep in class.
Sleep in class.
Sleep and write.
Mm.
My hand will be moving, but my eyes will completely be shut.
All right. Now we are going into the next important part. The pistil, megasporangium, ovule, and embryo sac. Okay. First, let's look into the picture. All right. Pictures first. Couples. What are couples? See here. Female reproductive organ consists of stigma, style, ovary attached to the thalamus. Syncarpus ovary, single. Okay, and this is multicarpillary ovary, where each and every carpels will be producing fruits. Example, grapes. Mango is different. Okay, mango is different. Grapes, one bunch. Each and every carpel will produce one. Okay, one small fruit like. Then, see here, this is the zooming of ovary. All right, ovary consists of where it is attached. This is known as stalk, isn't it? Hullium. It would be connected by funicell. Then this is the micropylar end from which the pollen tube enters. All right, micropylar end, micropyle. This pole is known as micropolar. The other end is known as chelasa. So this is known as chelasal pole. So to protect the embryo, there will be two layers, outer integuments and inner integuments. Inside, we'll be having the embryo, which is a 7-celled, 8-nucleate complex embryo. 3 cells will be here in the micropylar end, which in that one is egg cell. In between, there will be a central cell, which has two nucleus. And here there will be antipodals, 3 antipodals. Yeah, all right. That's what we'll be looking into next. See here, first of all, this one single microspore, megaspore mother cell will be divided into 2 dyad. Okay, then it again gets divided into 4. All right, tetrad, dyad, tetrad divided into 4. Then again, these 4 will be divided into 4 and 4, 8. 3 will be moving towards the chelasal end, known as antipodals, and 3 would be moving to the micropylar end, all right, which is known as egg apparatus, and the above one would be known as egg. The in-between central cell, two nuclei would connect and combine to form central cell, also known as polar nuclei. Sometimes we'll be looking into it. When we are looking into it, you'll be understanding. This will nourish the embryo, like coconut water. All right, nourish the embryo and become stronger and stronger. So this is the final stage. In chelasal end, we would be having 3 cells known as antipodals. This is known as egg apparatus. The first 2 would be synergids, and the above one is the egg cell, which is waiting for the pollen generative cell. We saw a generative cell, isn't it? In pollen grain. It will be waiting for pollen grain. This is the filiform apparatus. When the pollen tube connects here, the enzymes will be neutralized and slowly it moves to the egg and connects and form an embryo. All right, embryo. So each and every part we are going to work on. Look into it. All right, look into it. How, what happens, and how it is formed. So now you are okay with the diagram, isn't it?
Yes.
Now let's look into what happens in megasporangium. What is microsporogenesis? Micro, one we saw in pollen grain. The process of formation of pollen grains from pollen mother cell. Here, megasporogenesis, the process of formation of embryo from the megaspore, that is megaspore, mother cell. Okay. Gynoecium represents the female reproductive part of flower. The gynoecium may consist of single pistil, monocarpellary, or may have many pistil, one pistil, many carpellary, multicarpellary. When there are more than one pistil, may be fused together, syncarpous. See here, if there are only one single pistil, it is known as monocarpellary. When there are more than one, it is known as multicarpellary. In some places, multicarpels combine together, fuse together to form a single carpel, single pistil, known as syncarpous, or maybe free apocarpus. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as the landing platform of pollen grains. Landing platform, stigma. The style is the elongated, slender part beneath the stigma. The basal, bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you have studied in grade 11. Okay. Arising from placenta are the microsporangia, commonly called as ovules. Number of ovules in the ovary may be one, as in wheat, paddy, or mango, many, as in papaya, watermelon, and orchids. According to the placenta only, we'll be having large number of seeds too. All right. See, this wheat, paddy, and mango all have only one seed. Think about papaya, watermelon, or lemon. They are multicarpellary. All right, multicarpellary. Okay, megasporangium, also known as ovule. Let us familiarize ourselves with the structure of typical angiosperm ovule. The ovule is a small structure attached to the placenta by the means of stalk called funicule. Funicule. The body of the ovule fuses with funicule in the region called hilum. Hilum. Okay, hilum. Okay. Thus, hilum represents the junction between ovule and funicule. Each ovule has one or two protective envelope called integuments. When you open the hard shell of the mango seed, there is a hard shell, isn't it? The mango seed. The outer one, the hard one, is known as outer integument. If you cut, there will be a small thin layer protecting the seed.
Yeah. Real mango seed.
In that, we'll be having that green seed. So that is known as integuments.
I've been watching some gardening videos I saw.
All right.
They'll first take out there'll be one small opening there. They'll take a knife, cut open the hard shell, and then take out the seed, put it in the tissue, spray some water for a few weeks until a small root is sprouted.
Yeah.
And then plant it. That's all.
Gardening.
I'm planning on mine in the future.
Let's complete our 12th grade perfectly, and we'll be planning for all this gardening, extra new species. You can create those new species too.
For that, but isn't that having, like, how can I say? The stamen, it has, even if any foreign gamete comes also, the stamen will not accept, right?
Stigma.
The stigma won't accept it, right? So how can we create new?
By grafting and everything. There are different methods of asexual reproduction, isn't it? You can easily graft two rose plants of different colors into a new color, double color. Cut the stem into a wedge and another one like this.
What about other plants which does not have grafting and cutting?
Generally, these plants have these kind of things. Not in all plants. If you
No,
want to
some
produce,
plants, even if you do grafting also, it will not come. And even if you put some foreign pollen gamete also, it will not accept. The stigma won't accept.
yeah, because it contains different enzyme. The same kind of enzymes will be needed, or that is what we will be looking into that. We'll be coming across that when it comes to pollination. Since the atmosphere is dispersed with a lot of pollen grains, pollen grains of different species also come and takes with the stigma, but the reaction doesn't happen. The neutralization reaction doesn't happen, which can neutralize the inner cellulose and pectin and produce the pollen tube. We'll be coming across that. Here itself, we'll be coming across that. Okay. Integuments encircle the nucellus. Integument encircle the nucellus except at the tip where the small opening called micropyle is organized. Opposite to micropyla end is called chalasa, representing basal part of the ovule. Enclosed within the integuments is a mass of cells called as nucellus. Nucellus. Okay. Cells of nucellus have abundant reserve food material. Nucellus, this is very important. There, for pollen grains, it is tapetum. Here, this nucellus, this is the one which is going to nourish the embryo sac. Nucellus. Located in the nucellus is the embryo sac of female gametophyte. An ovule generally has a single embryo sac formed from megaspore. Megasporogenesis. The process of formation of megaspore from megaspore mother cell is called as megasporogenesis. Ovules generally differentiate a single megaspore mother cell, MMC. There it is PMC, pollen mother cell. Here MMC, which is megaspore mother cell. Generally differentiate a single megaspore mother cell in the micropyla region. See here, these cells are the nucellus. This one single cell. This is the megaspore mother cell. This is the nucellus. This nucellus will nourish the embryo. Nucellus will always nourish the embryo. We are going to read detail about this. Okay. It is the large cell containing dense cytoplasm. Like tapetum, this nucellus also has dense cytoplasm and prominent nucleus. The megaspore mother cell undergo mitotic division. Mitotic division. What is the importance of MMC undergoing meiosis? First, it undergo mitotic division, then it will undergo meiosis. Meiosis results in the production of 4 megaspore. First, MMC will undergo mitotic division, then it will undergo meiosis. Through meiosis only, one single nucleus is being distributed into 8 nucleus. All right. Through meiosis, it forms 4 megaspore. All right. Female gametophyte. In a majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develop into female gametophyte embryo sac. See here, there may be 3 out of 4 megaspores. Only one would be forming embryo, other will degenerate and give nourishment to it. Nourishment to that one generating embryo. All right. This method of embryo sac formation from a single megaspore is termed as monosporic development. From one MMC, a megaspore embryo sac is formed. This is known as monosporic development. Monosporic development. What will be the polydy of cells of the nucellus? MMC, the functional megaspore, and female gametophyte. So, nucellus
and functional megaspores, other three will give nourishment, high nourishment to the embryo sac. MMC, megaspore mother cell will form a tetrad, and which results in formation of one embryo sac, that is female gametophyte. Okay. Let us see in detail about what happens in embryo sac. After the mitotic division, it forms 4. From that 4, one is forming embryo sac, other 3 will degenerate, giving nourishment. Let us study the formation of embryo sacs in little more detail. The nucleus of the functional megaspore divide mitotically to form 2 nuclei, which move to opposite poles, forming the 2 nucleate embryo sac. 2 more sequential mitotic nuclear division result in 4 nucleate and later 8 nucleate stage of the embryo sac. It is of interest to note that the mitotic division are strictly free nuclear. That is, nuclear division are not followed immediately by cell wall formation. Not followed by cell wall formation. After the 8 nuclear stage, cell walls are laid down, leading to the organization of female gametophyte embryo sac. First, during this division, okay, during this mitotic division, it forms 2 forms 4 forms 8. At this stage, there will not be any cell wall formation at all. After completing this 8 nucleated stage, then only cell walls would be forming. All right. Observe the distribution of cell inside the embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. Remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the central cell. There is a characteristic distribution of cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have special cellular thickening at the micropylar tip called filiform apparatus, which plays an important role in guiding the pollen tubes into the synergids. 3 cells are at the chalazial end, known as antipodals. Filiform apparatus play an important role in guiding the pollen tubes into the synergids. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac matured as 8 nuclei and 7 cells. Someone is coming at this. Which one?
I found it. I'm underlining everything so that when I read it again, I'll be able to get it. And she's getting most of the questions from here.
Yeah. You'll be getting a lot of questions from you.
She's not using any specific guide. That's what I'm very scared of.
Whatever guide is, if you're going through the book in the right order, you won't be needing any guides. You can answer anything just like that.
Yeah, but they give extra information and give it in a simple manner, right? My chemistry guide, I gave money for my teacher. Actually, someone asked that guy for a discount, reduce the price. Okay.
Then there may be different carpels. It can be one pistil, single monocarpellary, or multicarpellary, or when the multicarpels are fused together, it is known as syncarpous ovary. All these carpels, multicarpels, pistils are fused together to form syncarpous ovary. One, right? The ovary where it is attached is known as funicule. All right. Funicule and ovary is separated by hilum. All right. Hilum. There are 2 ends, micropyle and chalaza. In the micropylar end,
there is an opening. All right, opening, which is known as micropyla pole. All right. Then there are two protective integuments. Outer one hard shell, and there is an inner another thin protective cell, inner integuments. Then we'll be having nucellus, which surrounds the embryo. Nucellus is a thick cytoplasm, prominent nucleus cell, which will nourish the embryo sac. All right. Formation of embryo cell. One megaspore mother cell produce 4 cells. All right. Tetrad. In that, 3 is degenerative and 1 is forming the embryo out of 4. Then from this end, from this next part, what happens? Mitotic division of megaspore mother cells. One cell is divided into 2. This is known as dyad, which has 2 cells. This further divides mitotically to form 4, and this further divides into 8. All right, 8. Here, each and every stage, the nucleus is not surrounded by cytoplasm, but slowly 3 cells, slowly the formation of cell walls forms, and 3 will be moving toward the chala end, known as antipodals. 3 will be moving towards the micropylar end. In that is known as egg apparatus. In that, the bottom 2 is known as synergids, and the one above is known as the egg. In between, there will be 2 nucleus, which combines to form central nuclei. These 2 nuclei combine to form 2 nuclei together to form a central cell, formed by one. They form one cell. All right. So at the end, we can see that the embryo sac is 8 nucleated, 7 cell. 1, 2, 3, 4, 5, 6, 7 cell. All right. This egg apparatus is placed near the filiform apparatus. Egg apparatus in filiform apparatus. This filiform apparatus will guide the pollen tube into the synergids, then to the egg cell. That is what would be happening in all these parts. Okay. Now I want you to look into the pollination. This is one amazing topic, pollination. Why so much pollen grain is produced? Why the megaspore mother cell produce only one? And why there are different locules, and everything we'll be understanding here. Pollination. Pollination. In the preceding section, you have learned that the male and female gametes in flowering plants are produced in the pollen grains and embryo sacs respectively. As both types of gametes are non-motile, they have to brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of the pistil is termed pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. You can list the possible external agents. What are the external agents?
Like birds, animals.
Okay.
Biotic and abiotic agents.
There are biotic and abiotic agents too. Let's look into it. Example, that bee pollen tablet. Bee. Bee is one of the major pollinating agents. All right. Then other insects like butterflies and moths and also a major pollinating agents. Then wind pollination is also possible. Then water pollination is also possible. then self-pollination inside. All right. So all these are possible kinds of pollination. Depending on the source of pollen, pollination can be divided into three types. One, autogamy. Let's look into the picture and then we can proceed. Autogamy. Then geitonogamy and xenogamy. All right. There are three types. And in autogamy itself, there are different types. We will look into it. Okay. We'll look into the different types of autogamy also. Okay. Picture A. Look into this picture. Then sunflower type. Then seismogamous flowers and celestogamous flowers. Okay. First one is self-pollinated. Second one is cross-pollinated. Then third one is celestogamous flowers. We will check into each and everything. Don't worry about it. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of the same flower. In a normal flower, which opens and exposes the anthers and the stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity. And also the anthers and stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, common pansy, oxalis, and Commelina produce two types of flowers. Chasmogamous flowers, which are similar to flowers of other species with exposed anthers and stigma, and celestogamous flowers, which do not open at all. In such flowers, the anthers and stigma lie close to each other. When anther dehisces in the flower buds, pollen grains come in contact with the stigma to affect pollination. Thus, celestogamous flowers are invariably autogamous, as there is no chance of cross-pollination landing on the stigma. Celestogamous flowers produce assured seed set, even in the absence of pollinators. Do you think that celestogamy is advantageous or disadvantageous to the plant? Why? Autogamy, pollination within the same flower. All right, same flower. A normal flower like hibiscus or anything, it is an open flower, isn't it? The pollen small stigmas or stamens are also just near to the stigma. In hibiscus, it will be surrounded around the stigma itself. All right. So that's what we have to understand. So in this, there are two types of autogamy. One is chasmogamous, where the flowers open. All right, open. In celestogamous, the flower does not open at all. So for celestogamous, the anthers and stigma lie close to each other. When pollen anthers dehisces the flower buds, so they will be having a set number of seeds. Everything will be pollinated. All right, all the seeds will be pollinated. Do you think celestogamous is advantageous or disadvantageous?
Advantage.
Advantage. Definitely it forms. And what are the disadvantages of celestogamous is, since it's pollinating within insect, new breeds, strong DNA production will not be there. Pollination within same plants will be producing same kind of offsprings and everything. It will not produce a bigger variety of different varieties, and strong varieties will not be easy to find. All right. Okay. Now, are you familiar with chasmogamous and celistogamous?
Yes.
All right. The flowers which open, all right. Pollination within the same flower, which is open flower. Celistogamous is it doesn't open at all. It doesn't flower at all. It will be closed. All right. The stigma, that is the anthers and the stigma will be closed by so that it will pollinate. Okay. Geitonogamy. Transfer of pollen grains from the anthers of stigma of another flower of the same plant. See here, geitonogamy. Let's think we have a male and female flower in a same plant. Or bisexual plant itself, pollen grain of one flower pollinates in another flower of the same plant. That is known as geitonogamy. Pollen grain from the anthers of the stigma, anthers or stigma of another flower of the same plant. Although geitonogamy is functionally cross-pollination involving pollinating agent, genetically it is similar to autogamy since the pollen grains come from the same plant. Xenogamy. Transfer of pollen grains from anther to the stigma of different plant. This is only due to cross-pollination. During the pollination, what's the word there? Read it.
Brings.
Brings. Bring. Okay. Genetically different types of pollen grains.
Pollen grains to the stigma.
To the stigma. Okay. Agents of pollination. Agents of pollination, like you told, it can be biotic or abiotic. Plants use two abiotic, wind and water. Abiotic, wind and water pollination are abiotic, and one biotic, animals. Agents to achieve pollination. Majority of plants use biotic agents for pollination. Only a small portion of plants use abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for this uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen grains, pollen when compared to the number of ovules available for the pollination. So, wind and water pollination, it is by chance, isn't it? There is an uncertainty. If there is only one pollen grain, it may or may not pollinate, isn't it? So it will be producing a large number. The same thing happens with tortoise and turtles too. Out of thousands of eggs which are laid by tortoise, hardly one or two, hardly around 10 or 20 would hatch and move to the sea. In that, only one or two would survive. So the turtles lay large number of eggs. Next. Okay. Pollination by wind is more common amongst abiotic pollination. Wind pollinations also require that the pollen grains are light. Pollen grains should be light and non-sticky so that they can be transported in wind current. They often possess well-exposed stamens so that the pollens are easily dispersed into wind currents. A large, often feathery stigma to easily trap the airborne pollen grains. Wind-pollinated flowers often have a single ovule in each ovary and enormous flowers packed into an inflorescence. Now you are familiar with inflorescence, okay? A familiar example is the corncob. The tassels you see are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall that water is a regular mode of transport for the male gametes among the lower plant groups, such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of need for water for the transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria hydrilla, which grows in fresh water, and several marine sea grasses such as Zostra. Not all aquatic plants use water for pollination. In the majority of aquatic plants such as water hyacinth and water lily, the flower emerge above the level of water and are pollinated by insect or wind, as in most of the land plants. In Vallisneria, the female flower reaches the surface of the water by the long stalk, and male flower or pollen grains are released on the surface of the water. They are carried passively by water currents. Some of them eventually reach the female flowers and the stigma. In another group of water-pollinated plants such as seagrasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grain in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by the mucilaginous covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? What do you think, Ma? It will be colorless and do not produce nectar.
It produces, right, nectar?
No, they don't produce. Nectars are produced for animal pollinated.
It's like a river, little river.
It is a mutual understanding. You will get food, I will get pollination. All right, you will get food and I will get pollination. That is what we do here. See, pollen grains are distributed and slowly, by chance, they will go and attach with the ovary. Majority of flowering plants use a range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds, and hummingbirds. Birds, they are small birds. They are not a big bird. Okay. Dispersal of seeds are done by a lot of birds. But this is for pollination. Sunbirds and hummingbird, which are small. Okay. And bats are the common pollinating agent. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Even large animals such as primates, lemurs, arboreal tree-dwelling rodents, or even reptiles, gecko lizard and garden lizard have been reported as pollinators in some species. Often, flowers of animal pollinated plants are especially adapted for particular species of animal. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Okay. Animals are attracted to flowers by color or fragrance. The flowers pollinated by flies and beetles secrete foul odor to attract these animals. From book.
Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small in number, flowers are clustered
Mm.
into an
Inflorescence.
inflorescence. Inflorescence to
Study from here, or make them
make the
conspicuous.
to
Mm. Animals are attracted to flowers by color and fragrance.
animals attracted by flower and fragrance.
The flowers,
The flowers
pollinated by flies and beetles, secrete foul odor to attract these animals. Then this part, next line. Okay, we will complete this.
No, I couldn't find that line.
Okay. To sustain animal visit, the flowers have to provide reward to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flowers, the animal visitor comes in contact with the anthers and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body come in contact with the stigma, it brings about pollination. Okay. In some species, floral rewards are in providing safe places to lay eggs. Safe places to lay eggs. My God. Lay eggs. An example is that of the tallest fir, Amorphophallus. Amorphophallus. The flower itself is 6 feet in height. The similar relationship exists between the species of moth and the plant of yucca, where both species, moth and the plant, cannot complete their life cycle without each other. Moth deposits its eggs in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larvae of the moth comes out of the egg as the seeds started developing. See here, yucca plant. Both cannot complete their life cycle without each other. Moths will lay their eggs in the flowers and the flowers get pollinated. After pollination, it develops, the ovary develops and produce seeds. When the seeds germinate, the moth would come out. Or else it won't come out at all. Okay. Why don't you observe some flowers of the following plants or any other available to you? Cucumber, mango, peepal, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animal visit them and whether they could be pollinators. You will have to patiently observe the flowers over a few days and at different times of the day. You could also try to see whether there is any correlation in the characteristics of a flower to the animal that visited. Carefully observe if any of the visitor come in contact with the anthers and the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or a nectar without bringing about pollination. Such floral visitors are referred to as pollen robbers or nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy your efforts. Outbreeding devices. There are some questions from this also. This paragraph is also very important. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower.
continued self-pollination result in inbreeding depression, like we told in celestogamous flowers. It is continuously getting self-pollinated. So inbreeding depression will be there. Okay. All right. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma become receptive, or stigma become receptive before the release of pollen. Okay. Margaret, what do you think about this outbreeding devices? Okay. What do you think about this outbreeding devices? If
Where
mm,
are these people coming from?
majority of flowering plants produce hermaphrodite. Okay. Hermaphrodite. Flowers and pollen grains are likely to come in contact with the same stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is to produce unisexual flowers. In both male and female flowers are present on the same plant, such as castor and maize, monoecious. Okay. If male and female flower are present in the same plant, it is known as monoecious. If they are present in different plants, it is dioecious, like papaya, right? Papaya has male and female, right? It prevents autogamy, but not geitonogamy. In several species such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, dioecious. This condition prevents both autogamy and geitonogamy. What is autogamy? What is geitonogamy? What is autogamy?
Autogamy means self
Within the same flower.
within the same flower.
Okay.
Within the same flower.
Same flower. If it opens, it is known as seismogamous.
Seismogamous.
If doesn't open at all, it is known as celestogamous.
Celestogamous.
Okay, celestogamous flowers. What is geitonogamy?
Same plant, different flower.
Same plant, different flower. Good. Xenogamy is totally different plant.
Different flower.
Different plant.
Flower and different plant. Different flower and different plant else.
What? Pollination from one plant to another plant. Okay. Different plant, different flower. It means totally papaya and banana, like that. Okay. All right. Okay. Some like papaya have female plant, male plant. There you see. How and all this is possible?
Papaya has papaya. We are having papaya.
How outbreeding devices are possible? See here, the receptivity of stigma and the release of pollen grains may be different. So self-pollination, that is autogamy, may not be possible. All right. One pistil may be matured faster and anthers will be slow to mature. So it will prevent self-pollination. Other ways is you can prevent it by stopping the movement of movement of pollen tube in stigma. There is style, isn't it? Pollen tube should be moving down. If you stop that, automatically it will get stopped. All right? Or else, any other way? To prevent inbreeding in, okay, to prevent inbreeding
Third
is
device. Oh my god, I didn't see it.
okay. All right. Generally, this
Another device to prevent self-pollination is the production of
unisexual flowers, male flower and female flower. Okay, male flower and female flower in same plant, like castor and maize. Monoecious, known as monoecious. It can prevent autogamy but not geitonogamy, because same plant itself has the both male and female flowers. And some places, some plants, papaya, male and female flowers of different plants. Okay, right? Okay, now we are coming to this. This is what you asked for, pollen pistil interaction. That is, other different pollen grains can come and stick in the different stigma, isn't it? That one you asked about. That's what pollen pistil interaction is all about. All right. Can you proceed then?
My topic is.
Pollination does not guarantee the transfer of right type of pollen, compatible pollen of the same species as the stigma. Often pollen of wrong type, either from other species or from the same plant, if it's self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type or wrong type, compatible or incompatible. That is, it may be from the same flower, same type of flower, but it may be incompatible. It won't germinate. The viability is finished. Those also can be possible, and from other plants, other different flower, totally different flower, that is also possible. So generally, this stigma has the ability to recognize the pollen, whether it is right type, compatible, or of the wrong type. If it's right type, the pistil accepts the pollen, promotes post-pollination event, that is pollen tube formation. See here how the pollen tube is getting formed. These are pollen grains, and pollen tube is getting formed and going inside the stigma. It will be very, very minute, isn't it? Pollen grains, after getting into the stigma, it slowly from the germ pore produce this pollen tube with the generative cell nucleus. All right. So it will not be one or two. It will be many will be traveling. Which one is faster and first to penetrate the filiform apparatus will get the go. All other strands has to stay just like that. Okay. See here, this many is getting, these are incompatibles. So it doesn't produce, it doesn't allow to produce pollen tube at all. Some will be slow. Some would have deposited first itself. This one would be the first to go and get into the micropylar end and find the desired cell. This filiform apparatus which we have, we will be going through those. There, this filiform apparatus, actually this is also chemical. Slowly it will disintegrate. This filiform apparatus and this walls of the pollen tube will disintegrate, and it will produce the, finally release the pollen, the male gamete. Okay, male gamete will be released to the cell. This is how this happens. Okay. Pollen grains germinate on the stigma first, then pollen tube growing through the style, then pistil showing path of pollen tube growth. Enlarged view of an egg apparatus showing entry of pollen tube in the synergid, this one. Then E, discharge of male gamete into the synergid and movement of sperm, one into the egg and the other into the central cell. See here? So the movement of sperm will be one to the egg, other would be to the center cell, forming triploid nucleus. You will be getting across that. In the next part, you will be getting across that post-fertilization event. So that's what we are viewing here. The pistil accepts the pollen and promote the post-pollen events that lead to fertilization. If the pollen tube is of wrong type, the pistil rejects the pollen. Wrong type, pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in this style. This ability of pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between pollen grain and pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. No, my got disconnected and again reconnected. It's like a chemical interaction. See here, acid and base will react and neutralize. Like that, the stigma as a chemical and the pollen grain as a chemical. When both interact, they will disintegrate and the tube is formed. It will allow the formation of tube. Or else, the reaction doesn't take place at all. Chemical reaction doesn't take place at all. So it is known as a dialogue between pollen and the pistil stigma. All right. So that you are right kind. Okay, I will accept it. We will grow. All right. In filiform apparatus, there will be different chemical which will neutralize the pollen tube, the membrane of the pollen tube, and help it to release the cells. One cell, the generative cell, will go and attach with the egg, and the vegetative cell will go and attach with the central cell, and form a triploid. Triple nucleated. Okay, triple nucleated. It's not a triploid, triple nucleated cell, which will again enrich and give nutrition to the growing embryo. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to the recognition followed by acceptance and rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce pollen tube through one of the germ pores. The contents of the pollen grain move into pollen tube. Pollen tube grows through the tissue of the stigma and style and reaches the ovary. You would recall that in some plant, pollen grains are shed at two cell condition, a vegetative cell and a generative cell. In such plant, generative cell divides and forms two male gametes during the growth of the pollen tube in the stigma. In plants which shed pollen in a three-cell condition, pollen tubes carry two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergid guides the entry of pollen tube. All these events from pollen deposition on the stigma until pollen tube enters the ovule are referred to as pollen pistil interaction. All right. See here. pollen tube would reach the ovule through the micropylar end. Then enters the synergid through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergid guide the entry of the pollen tube. That is, it will slowly neutralize and decrease the pollen tube. The chemical in the pollen tube, the tube, will neutralize the filiform. Automatically, which way it will neutralize? It will be towards the way of the egg. Okay. So that's known as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition. What is inhibition? What is meant by inhibition?
Promotion.
Promotion is okay. You are allowed.
Inhibition means
It will stop. No, you are not allowed. It's like a bouncer in any party.
If it's useful, it will allow.
Yeah. Or else it will not allow the unwanted peoples. The knowledge gained in this area would help the plant breeder to manipulate in manipulating pollen pistil interaction, even in incompatible pollination to get desired hybrids. Okay, if you understand this process only and in a rightly controlled condition, then only you can go in for another breed cross pollination. Accepting this condition. See, the stigma should accept, the style should accept, and the filiform apparatus should accept, or else it will be stopped, arrested in between. One, stigma accepted, it will start growing, but style will not accept. It will stop it. Okay, style also accepted. The filiform apparatus will stop it. All right. So that's how it can be done. You can easily study pollen germination by dusting some pollen tube flowers such as pea, chicken pea, or crotalaria, balsam, and vinca on a glass slide containing a drop of sugar solution, about 10%. About 15 to 30 minutes, observe the slide under the low power lens in the microscope. You are likely to see pollen tubes coming out of the pollen grains. As you shall learn in the chapter on plant breeding, chapter 9, a breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization. This is also very important. These are the important questions you can get. Okay. Artificial hybridization. In one of the major approaches of crop improvement program, in such crop crossing experiment, it is important to make sure only desired pollen grains are used for pollination, and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging technique. We need a desired superior quality fruit. So we need right combination of pollen grain to interact with the pistil. All right. So what and all can be done is to stop self-pollination. We can do emasculation or bagging, followed by bagging. In the female parent, bearer bisexual flowers, removal of anthers from the flower bud before the anther dehisces using a pair of forceps is necessary. This step is referred to as emasculation. That is, before the anther developed and dehisce, broke down and release the pollen, the anther should be cut with the forceps. Remove it completely. All right. First, emasculated flowers have to be covered with bag of suitable size, generally made up of butter paper, and prevent contamination of the stigma with unwanted pollen. After emasculation, now the flower is getting all the stamens are removed before they mature at all. Only stigma is there. Now we have to cover that stigma with butter paper so that we can prevent the contamination of other pollen grains. Right? That is known as bagging. Okay, that is known as bagging, butter paper, to prevent contamination. When the stigma of the bag flower attains receptivity, mature pollen grains collected from the anther of the male parent are dusted on the stigma, and the flower are rebagged, and the fruit allowed to develop. Now, after checking that whether the stigma is being receptive now, we have to sprinkle it with the desired pollen grains and again rebag it and left it alone so that it will form pollen tubes, mature, and bear fruits. All right. Okay. If the female parent produces unisexual flowers, there is no need to emasculation. The female flower bud are bagged before the flower open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebag. If it's a unisexual flower, what we will do? We don't have to emasculate because it will not be having any male androecium. There will not be any stamens. So you just have to bag it. When it attains receptivity, use desirable pollen and again rebag it so that it will grow. Before the bud open itself, they will be packing it. Okay. Okay. Double fertilization. Okay. How double fertilization is possible? This is also an important question, double fertilization. After entering one of the synergids, the pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gamete moves towards the egg and fuses with its nucleus, thus completing the syngamy. What is syngamy? It will not be here in this lesson.
I thought I missed it.
Syngamy is the process of it came in first lesson. All right, it came in first lesson. Syngamy is the process of mating of male and female gamete, egg and sperm. All right, that is known as syngamy. This results in the formation of diploid cell, zygote. You want to write it? Okay.
I forget. Now I'll remember, but next time when I read it, I'll not remember.
Okay. After syngamy, what will be the result? The formation of zygote. All right. The other male gamete move towards the two polar nuclei located in the central cell and fuses with them to produce triploid primary endosperm nucleus. Triploid. Why? Already in central cell there will be two nuclei, and one vegetative nuclei will be going and joining there, so it becomes triploid. Okay, triploid primary endosperm nucleus, also known as PEN. Primary endosperm nucleus. As this involves the fusion of three haploid nuclei, it is termed as triple fusion. Since two types of fusion and syngamy and triple fusion take place in the embryo sac, the phenomenon is termed as double fertilization, an event unique to flowering plant. The central cell after triple fusion becomes primary endosperm cell, and develops into endosperm, while the zygote develops into embryo, Okay, see here, synergid is here, It is not upside down, it is straight. Okay. So the pollen tube interacts and commit and releases nucleus. This nucleus will combine here, 1. And the generative cell goes and forms 3N primary endosperm nucleus, pen. Okay, pen. And after the zygote is getting nourished and undergo meiosis division, this primary endosperm nucleus, pen, will be the nourishment for the zygote. All right. Okay. Post-fertilization structure. Post-fertilization structure and events. Okay. Following double fertilization, event of endosperm and embryo development, maturation of ovule or ovules into seed or seeds, and ovary into fruit are collectively termed as post-fertilization events. Post-fertilization event. Endosperm. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and form a triploid endosperm tissue. The cells of this tissue are filled with reserve food material and are used for the nutrition of the developing embryo. In the most common type of endosperm development, pen undergoes successive nuclear division to give rise to free nuclei. This stage of the endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. The tender coconut which we drink, the liquid which we drink, is the free nucleus. Slowly, as it matures, it becomes cellular nuclei, and the thick kernel which we eat,
The white
that white one will be found. But the first one would be only liquid free nuclei endosperm. It will slowly convert into that white kernel. Okay. What I would do is, my mom would be cutting. Slowly I will pick and swallow it and run away before she asks.
Won't it get stuck?
No, they will be doing that.
Oh, it will be grinded.
Grinded. I will take it. It will be tastier, that one, after grinding. Endosperm may either be completely consumed by the developing embryo. Example, pea groundnut beans. This endosperm cells, that free nuclear endosperms, can be completely disintegrated. That is, it is getting nourished. That's why peas, groundnut, and beans are really, really nutritious. All right. Before seeds maturation, or it may persist in the mature seeds, castor and coconut. In coconut and all, even after it is maturing, we have some water, isn't it, inside? That will be there, just like castor or coconut, and be used up during seed germination. Split open some seeds of castor, peas, beans, groundnut, fruit of coconut, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Embryo. Embryo. It is also another important part. We are nearing to the conclusion of the lessons. Okay, only one more part is there after completing this. Okay, embryo, then seeds. Embryo. Look into this diagram. Mine is cut down a sentence.
Why is this?
Maybe while downloading it got this. See this?
Yeah.
Okay.
Important.
All right. After this, we can see
Oh no, that's not required.
which one. Oh, this one is not required now. All right. Primule and radical. This primule will grow up into shoot system. This one into root system. All right. Number one, a typical dicot embryo. There are two cotyledons, one cotyledon, two cotyledons. All right. If it's a normal monocotyledon, there will be only one. Since it's dicotyledon, we have two lobes. All right. Primule will move into shoot system, and radical will move into root system, which has a root cap. What is the use of root cap? What is the use of root cap?
This
See, here will be the meristematic tissues. The tissues the cells which will undergo rapid divisions, mitotic division to form numerous cells. That cell should be protected. So as it grows, it will be moving ascended. That cell should not be damaged. So there will be a root cap to protect those meristematic cells. What is meristematic cell? The cells which are involved in growth. All right. There may be lateral and horizontal also. Okay. B. LS of an embryo of the grass. See here, what and all we have. Here also we'll be having a
root cap.
radical root cap, and this one would be the shoot system. This will shoot up, this one will shoot down. All right. This is scutellum. We'll be looking into that. When we are working here, we'll be looking into that. Then coleoptile, which will protect the grass, sharpness of the grass. Even with the grass blade, if you cut it with a basic method, you can easily damage skin also. We may say grass is soft as sensitive. Take a grass and perfectly place it in one side and fish it. You can easily cut. That much hard it will be in that way. All right. So this is epiblast. This one will be the shoot apex. All right. That one we are going to read. Then seed, type of seeds. We are going to complete the lesson soon then. Let's proceed. Okay, embryo. Embryo develops at the micropylar end of the embryo sac, because that's where we had that zygote, egg and male gamete getting fused. Most zygote divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seed differ greatly, the early stage of embryo development, embryogeny, are similar in both monocotyledon and dicotyledon. 2.13, this is the stage of embryogeny in a dicotyledonous embryo. The zygote gives rise to proembryo and subsequently to globular and heart-shaped mature embryo. A typical dicotyledon embryo consists of embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledon is epicotyle, which terminates with the primule or stem tip. The cylindrical portion below the level of cotyledon is hypocotyle, that terminates at the lower end of the radicle or root tip. The root tip is covered with the root cap. Embryos of monocotyledon possesses only one cotyledon. In the grass family, the cotyledon is called scutellum. The grass family, the cotyledon is known as scutellum. that is situated toward one side of the lateral of the embryonal axis. At the lower end of the embryonal axis at the lower end, the embryonal axis as the radicle and the root cap enclosed in an undifferentiated sheet called coleorea. Okay, coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and the few leaf primordia enclosed in the hollow foliar structure, coleoptile. Okay. Soak a few seeds in water, say wheat, maize, peas, chickpeas, groundnut overnight. Then split the seed and observe the various parts of the embryo and the seed. What's there in the picture will be explained. Okay. What's there in the picture will be explained. This will become shoot system, which has two cotyledons, this hypocotyl, which has the radicle and the root cap. And for a grass, a monocotyledon, the shoot system generally consists of the scutellum. All right. The cotyledon would be the scutellum. And this is the shoot apex, which is covered by coleoptile. All right. And the middle, which is the part where it touches the ground. All right. Epiblast. One would be moving upwards, shoot system, and another downwards. All right. Root system, radicle, root cap, and coleorhiza, which covers this overall area and protect it. That's what about embryo. And the embryo develops into seeds. Embryo develops into seeds. In angiosperm, the seed is the final product of sexual reproduction. It is often described as fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coats. That coats are the integuments, okay? Cotyledons and the embryo axis. The cotyledon figure of the embryo is a simple structure, generally thick and swollen due to the storage of food reserves, as in legumes. Legumes means all these pea plants, beans, these type of plants are known as legumes. All right, legumes. Mature seeds may be non-albuminous or albuminous.
Pea plants are
The pea type of plants, no? All those plants, chena, all these are legumes. Okay, that plant variety. Okay. The mature seeds may be non-albuminous or albuminous.
no.
Hmm?
Ex-albuminous.
X. AX. Ex-albuminous. Ex-albuminous. Non-albuminous seeds have no residue endosperm, as it is completely consumed during the embryo development. Example, pea, groundnut, maize, all these are completely consumed endosperm. All right. Then albuminous seeds retain the part of endosperm as what's the term?
As it is not completed.
As it is not completely used up during the embryo development. Example, wheat, maize, barley, castor, coconut. Coconut also can be added with this. Occasionally in some seeds such as black pepper and beet, remnants of nucellus are also present. What is nucellus, ma? What is nucellus?
The center of the
The embryo,
Center of the
no, the megaspore mother cell, MMC, is covered by a thick cells which have dense cytoplasm. For an ovary, it is nucellus. For pollen grain, it is tapetum. All right. Okay. Are also persistent in some black pepper and beet. This residual permanent nucellus is perisperm. Perisperm layer. It forms a layer. Residual forms a layer. We will know what is perisperm soon. Integuments of ovules harden as tough protective seed coats. The micropyle remains as a small, what's the word? Yes. Micropyle remain
small
as
pore.
Small pore only. Okay. Small pore in the seed coat. This facilitates entry of oxygen and water into the seeds during germination. As the seed matures, its water content is reduced and the seed becomes relatively dry, 10 to 15%. 10 to 15% dry. By mass, okay. General metabolic activities of embryo slow down. Embryo may enter the state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. As ovules mature into seeds, the ovary develop into a fruit. The transformation of ovule into seeds and ovary into fruit proceeds simultaneously. Both formation of seeds and fruit will proceed simultaneously. The wall of the ovary develops into a wall of fruit called pericarp. Pericarp. The fruit may be fleshy as in guava, orange, mango, or may be dry as in groundnut, mustard. Many fruits have developed mechanism for dispersal of seeds. Recall the classification of fruits and the dispersal mechanism that you have studied in the earlier classes. Dispersal mechanism. Is there any relation between number of ovules in an ovary and the number of seeds present in the fruit? How many ovules are there? That many seeds will be. So mango plant have how many ovules?
One.
Only one.
Only one.
What about grape?
Many.
Yeah, many different number of ovules will be there. Seeds will be there. In most plant, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. After the formation of embryo, formation of zygote, slowly the style, stigma, anthers, corolla, everything will fall off. All right. However, in few species such as apple, strawberry, cashew, the thalamus also contributes to fruit formation. Such are known as false fruit. The fruit which we eat in apple is the thalamus, not the original fruit. This one is the original fruit.
Original.
Yeah.
This is false.
Yeah, false fruit. Thalamus. Most fruits, however, develop only from ovary and are called true fruits. Although in most of the species, fruits are the result of fertilization, there are few species, cotyledons, two cotyledons, and at the micropyle there will be a small opening which will help in movement of oxygen. All right. Seed coat. How it develops. Okay, seed coat, endosperm, and cotyledons. And slowly the cotyledon will be breaking open and forming. Okay. These are false fruit. This pericarp, the pericarp which we spoke about, actually it will be in 11th standard. Okay, 11th standard. Pericarp is made up of three parts: epicarp, endocarp, and mesocarp. All right, epi, endo, meso. All these three combine to form pericarp. In berries, endocarp and mesocarp would be fused. Example, tomato. All right, the edible part which we eat. The outside, the thin layer is the epicarp. Endocarp and mesocarp will be fused together in tomatoes. Okay. There are few species in which fruit develops without fertilization. Such fruits are called parthenocarpic fruit. They doesn't need fertilization at all. They are known as parthenocarpic fruit. Banana is one such example. Parthenocarpy can be induced through application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperms. Firstly, since the reproductive process such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitats and help the species to colonize in other areas. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combination leading to variation. New genetic combination leading to variation. Seed is the basic of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to raise the crop for the next season. Can you imagine agriculture in the absence of seeds or in the presence of seed which germinates straight away soon after formation and cannot be stored? How long do seeds remain alive after they are dispersed? The period again varies greatly in some species. Lost viability within few months.
I know what I did.
Oh, internet getting off and on. Okay. Lose viability within few months. Seeds of large number of species have for several years. Some seeds remain alive for hundreds of years. There are several records of very old yet viable Lupinus arcticus excavated from Arctic tundra. The seed germinated and flowed after 10,000 years. A recent record of 2,000-year-old viable seed is of date palm, phoenix, datilifera, discovered during the after completion. After completion, a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of several flowering plants, asking the following question: how many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on the tree, and so on? Can you think of some plant in which fruit contain very large number of seeds? Orchid fruits are one such. What's that, ma? One such category, and each fruit contains thousands of tiny seeds. Similar in the case of fruits of some parasitic species such as Orobanchi, and Striga. Have you seen a tiny seed of fucus? How large is the tree of fucus developed from a tiny seed? How many billions of seed does each fucus tree produce? Can you imagine any of other example in which such a tiny structure can produce such a large biomass over the year? Here, the fucus tree, Attipalan Sulong in Tamil. Fig. Okay.
I'm drinking.
The fruit will be like a conical shape, almost like a pear. All right. That one will be produced, the seed will be minute, just like a grain of sand. Just like a grain of sand, it will be very, very minute. All right. But the size of the tree is enormous. All right. Only one page is balance in this lesson. We have completed the almost oral lesson. All right. Okay. You need a small break before we start this.
I'll just wash my face and come.
Okay. Come on. Apomixis and polyembryony. Although seeds in general are the product of fertilization, a few flowering plants, such as some species of Asteraceae and grass, have evolved a special mechanism to produce seeds without fertilization called apomixis. Asteraceae, grass. Asteraceae, all the sunflower type of flowers, no, they are from Asteraceae. Sunflower type Asteraceae. All right.
Handwriting.
You can understand?
Yeah, I can understand because this is my handwriting.
Okay.
Nobody will understand. But
Thus, apomixis is what is fruit production without fertilization called? That is apomixis. Okay, without fertilization. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac start dividing. Nucellar will start dividing. Okay, nucellar will start dividing, protrude into the embryo sac and develops into embryo. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Okay. Take out some seeds of orange and squeeze them. Observe that many embryos of some different size and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clones? Okay. Yeah, they are developing without fertilization. They can be called as clones. Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids have tremendously increased productivity. Hybrids. One of the problems of hybrid is that hybrid seeds have to be produced every year. If the seed collected from hybrids are sown, the plant is the progeny in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seed is costly, and hence the cost of the hybrid seeds become too expensive for the farmers. If these hybrids are made into apomix, there is no segregation of characters in the hybrid progeny. Then the farmer can keep on using the hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, Active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties. Wow. Okay. Awesome.